Alright, so we're going to start off now with chapter 21, which is our chapter on plant form, different types of cells, different tissues, and plant organs. Now, most of the chapter is going to be in the lecture format that you're going to see right here, but once we get toward the end of this, I'm also going to post a section that deals with plant transport, because I know that's in the chapter as well. Again, I don't really like the book PowerPoints. I like to use my own. So it's going to be a little bit disorganized. It's not going to look exactly the same as what it does in your book, but it certainly is the same information, maybe with some hopefully better pictures, or some of the same pictures at least, and a bit more explanation as well to go along with them. So in terms of plant organization, there's really only about two different organ systems, right? We talk about animals and all the different organ systems in at least vertebrate animals, like there's 11 of them. In plants, there's only 2. Now, it doesn't mean plants are any less complicated than animals, but they're set up differently, right? So the first organ system we find in a plant is the root system. And the root system is there to help, obviously, anchor the plant to the ground, but it's also there for storage of things. There are plants that use their roots to store carbohydrates. There are plants that use their roots to spread asexually or clonally throughout their environment. Obviously picking up water and nutrients from the soil as well, things like nitrogen and phosphorus, some of the plant nutrients you find in the chapter. Those, for the most part, are also absorbed through the roots. Really, the only thing that's not absorbed through the roots is carbon dioxide, which comes in through the leaves, obviously, for photosynthesis. But a lot of functions there for the roots. The shoot system is everything above ground. So everything including the stem, the leaves, flowers for plants that produce flowers. And not all plants do. There are 3, actually, groups of plants, big ones, that don't produce flowers. But those could be on a shoot system as well. And we'll take a look at some of the different types of leaves, different modifications of leaves, different functions of leaves as well as for stems. We're going to take a look at different modifications for all of these. And so just on this picture here, this is a picture of a bean plant, and we can see some of the things labeled on here that are important. A node is a point on the stem where something branches off. So right here there's a node, right where the arrow is a node, where you have this little branch coming off of the stem. An internode is the distance between 2 nodes. So if there's a node here and a node here, the distance between the 2 then is the internode. Leaves are sometimes a bit more complicated than what you might think they are. Leaves can be either compound or simple, and they come in all different shapes and sizes, of course, so lots of variability in leaves. And then we'll see terms like axil. So if we look at the angle between the branch and the stem, there's this little angle right here, and that angle is called an axil. And if you see a bud in that axil, that bud is called an axillary bud. Not an auxiliary bud, that would be something different, but an axillary bud because it appears in the axil of that plant. As far as tissue types, there's only 3. Again, animals have plants beat there as well, because in animals we have 4 different tissue types, of course, epithelial, connective, muscle, and nervous. In plants, there are only 3 tissue types. That being said, those tissue types can consist of multiple different kinds of cells. So different kinds of tissues we find in plants include dermal tissue, which is kind of the plant equivalent to what epithelial tissue is, although it serves kind of a different function. There is ground tissue, which is typically the most abundant tissue in any plant is ground tissue, and that's where you find a lot of storage function and photosynthetic function, secretion of things, just making up the overall body of the plant in many cases happens with ground tissue. And then vascular tissue, which sounds like cardiovascular. Vascular tissue is what's used for transport inside of a plant. So we'll take a look at different types of vascular tissue, as well as how things like water and carbohydrates move through the plant as we go along here. Now, one of the things that's a bit different from what plants are as opposed to what animals are is that the tissue systems go from organ system to organ system. So there's a connection between, say, the vascular tissue in the roots and the vascular tissue in the stem. There's a connection between dermal tissue in the roots and dermal tissue in the stem. So there's this connectedness above ground and below ground of these 3 different cell or tissue types that's a little bit different than how you would kind of picture or imagine things in an animal. If we look at different types of plant cells, right, there's different kinds here. We can look at the size of vacuoles. They have different types of plant cells, and really there's only about 3 different types. The size of the vacuole is one defining feature of the kind of plant you might be looking at. You can look at whether the cells are living at maturity or not. So for the first 2 cell types, we'll call those parenchyma cells and cholenchyma cells. Those 2 cells are both living at maturity. If we look at the 3rd type of cell, and you'll see these spelled out here coming up here on the slides, sclerenchyma cells are always dead at maturity. So it's not until after they've died that a sclerenchyma cell becomes functional. And typically for those cells, they're involved in structure and strength and those kinds of things. So they don't really need to be alive at maturity. We can also look at the thickness of the cell walls. Parenchyma cells have very thin cell walls, which makes them very inexpensive to produce. Collenchyma cells have thicker cell walls that are flexible. That makes them a bit more expensive to produce, but not nearly as expensive as the third type of cell, which is a sclerenchyma cell. Very, very thick, very heavily reinforced cell walls that give them a lot of structural strength, especially, like we said before, after they're dead. And that's kind of their job, is to be strong for the body of the plant. One of the other things that we're going to look at in plants that's extremely different from what animals do is plant growth. Right? So plants, kind of the easiest, simplest way to think about this or to say this is that plants only grow at the tips. They only grow at the tips of the shoot, and they only grow at the tips of the roots. Right? These cells in these regions of the plant are kind of like stem cells. They are called meristematic cells. And when a meristematic cell divides, when it goes through mitosis, it produces another meristematic cell, and then some type of differentiated cell. Maybe this is a vascular tissue cell. And then this meristematic cell divides again through mitosis to produce yet another meristematic cell and another differentiated cell. Maybe this one's a ground tissue cell. And then the other meristematic cell divides again to produce another meristematic cell and another differentiated cell. And this goes on more or less indefinitely in the meristem. So in terms of plant growth, plants really never ever stop growing because they always have the supply of meristematic cells. If you think back to our chapter on animal reproduction, we talked about spermatogenesis and how spermatogonia, when they divide, they give rise to another spermatogonium and also then a primary spermatocyte. So males, as they produce sperm cells, always have a supply of sperm cells going forward. Kind of the same thing in a meristematic system. You got this continuation of the meristem cell line, even though you also have differentiation at the same time. But what this does is that it ensures that you always have a supply of cells that can divide moving forward. Meristems, like I said, are located in the tips of the shoots, the tips of the roots. Those meristems are what we call primary meristems, and they're the ones that are going to let both the roots and shoot get longer. They result in an increase in length as they grow. The other type of meristem is called a lateral meristem, and that kind of meristem allows plants to get bigger around. So they increase in diameter as growth goes on as a result of secondary growth. So apical meristems are the ones at the tips, because apical refers to apex, which means like the top or the tip of something. And meristem refers to these cell types that are kind of like stem cells. Apical meristems are at the tip of the shoot, tip of the root. Lateral meristems in plants that do this, in plants that get bigger in diameter, are found all along the length of the stem and along the lengths of the roots. But again, not all plants do this because some plants only do primary growth. The kinds of plants that do secondary growth are woody plants, trees, that have rings that they produce. Those rings are produced by lateral meristems. All right, so we'll take a look at the apical meristems first in the stems and the roots. We take a look here in this picture. We've got the shoot tip, both unstained and stained, just for kind of clarity. And then also down here a root tip, both unstained and stained, to show you the different cell types. So up here in this picture, all these green cells out here are dermal cells. And all the light orange cells here are ground tissue cells. The dark orange cells, of course, then are the vascular tissue cells, because that's the only tissue types they have. Same thing holds true in the roots. We've got dermal cells on the outside. We've got ground tissue cells here in the middle. And then we've got vascular tissue cells all the way there in the center. So those are what we call the primary plant body. They're going to produce the primary tissues. In this picture up here, the apical meristem is going to be right up here. This is the very, very tip top of the stem. This is the first baby leaf, the very next baby leaf in line that's been produced. This is the second youngest and third youngest and fourth youngest, and eventually, as you were to move down the tip of, or the down through the shoot, you would see bigger and bigger and bigger leaves. Over down here, we've got the apical meristem right about there. We have something called a root cap out here. The root cap is involved in protecting the apical meristem, but the apical meristem here is the one that's actually dividing and adding cells toward the tip of the root. So they're both very, very delicate areas. They're both very sensitive areas, and they're both very important areas to protect because they are so delicate and sensitive. Very important areas. If the apical meristem is lost from a shoot or from a root, that root will never get any longer. That shoot will never get any longer. It may be possible to branch off a second shoot or a second root and have that one increase in length, but if the apical meristem is gone, then you have no more primary growth at that point. Right, so again, we talked about the root cap. That's involved in the soil because soil is a very abrasive substance. If you don't have some sort of physical protection of the apical meristem, it would be damaged and rendered inoperable very, very quickly. In the shoot, air is obviously a much less abrasive substance than soil, so you don't need to have as much protection in the shoots. But what you do have up here are what are called leaf primordia, these little bumps here that eventually turn into leaves. They're going to be on either side of the apical meristem, again, to help protect it from physical damage. All right, so these apical meristems, when they are dividing, when these cells are dividing, again, we've got up here an apical meristem. We've got the two youngest leaves are right here. These are leaf primordia. We've got the next two oldest leaves here, and the oldest leaves in this picture are these ones here. So they kind of go off the picture because they're so big. But if you were to look inside of this, as these cells are dividing, you're going to be producing three primary tissue types. The tissue type all the way on the outside is called protoderm. This outermost layer is called protoderm. Proto means before, derm is referring to dermis, so this is the pre-dermal tissue produced by the plant. We also have a procambium. Procambium is all the way in the middle. Procambium is going to give rise eventually to vascular tissue. These cells eventually differentiate into vascular tissue. And then sandwiched between the protoderm and procambium is the ground meristem, which is right in here, and that's going to give rise to ground tissue. So these three are like the precursors to the main primary tissue types we talked about before, the dermal tissue, the vascular tissue, and the ground tissue. Other plants have what are called intercalary meristems, which are kind of located along the stem in rings. So think about corn. Think about a corn stalk. And you know how there are those kind of rings that go around the stem at various intervals as you go up the stem? Those are different types of meristems that help with primary growth. They allow corn to grow really, really fast. In fact, most grasses, and corn is a grass, most grasses have something like an intercalary meristem to help them grow really fast, which is why you have to cut your grass once a week in the summer when it's growing quickly. There are meristems all along the length of them that help them grow and get longer. So they make the internodes longer very, very quickly. Lateral meristems, again, are what produce secondary growth, and secondary growth, again, is growth in diameter. Not all plants do this, but for plants that do, secondary growth from lateral meristems results in the production of the secondary plant body, which in most cases is made up of what's called secondary xylem, secondary phloem, and cork cambium. So in woody plants, there are two types of lateral meristems. The cork cambium is one. This is the part that produces the bark. So the cork cambium, I don't know if you can see in this picture here, the cork cambium is going to be almost all the way on the outside of this plant. Right? And this picture here is showing you all the way at the tip what the stem is going to look like. Further down in the stem, where the stem is much older, what that's going to look like. Pretty high up in the roots, so a more mature root's going to take on this appearance. And then all the way down at the tip, the roots are going to take on that appearance like that. Right, so the cork cambium is producing bark. The vascular cambium is producing new water-conducting cells and carbohydrate-conducting cells inside the stem and roots. Because as a plant grows in diameter, as secondary growth continues, what occurs is that the water-conducting and food-conducting parts of the stem or roots get damaged and get broken. So these two cambia, cork and vascular cambium, are going to help replace the water-conducting cells and the carbohydrate-conducting cells. So starting off in a first-year stem, or in the case of this picture up here, there are primary xylem. Xylem are used to conduct water, and there are primary phloem cells. These guys are what conduct carbohydrates. Typically, xylem is carrying water from the roots up the stem to the leaves, and phloem is carrying carbohydrates and sugar from the leaves down the stem toward the roots. They're kind of going in different directions. One's carrying water up, the other one's carrying carbohydrates down. Now, as time goes on, if you were to really zoom in on this picture here, what you would see is between this blue area of primary xylem and red area of primary phloem, there's going to be a layer of what's called vascular cambium. And as that vascular cambium divides, it's going to separate the primary xylem from primary phloem. So this blue area and red area are actually going to become more and more separate over time. And so the green layer here, which is the vascular cambium, we'll see a bigger picture in a second, but this vascular cambium here is going to add secondary xylem on the inside and secondary phloem on the outside of itself. So it's always producing new water-conducting tissues and new carbohydrate-conducting tissues all the time as long as it's alive. And as it does that then, as it adds new layers of cells, that's how the stem gets bigger around. That's how it increases in diameter, is by the addition of cells from primarily the vascular cambium. Right, so secondary xylem is water-conducting. It's what's added after a period of time when the tree starts to age. It's also the main part of the wood. So if you cut down a tree, or if you see a piece of firewood that's got rings in it, those rings are year after year after year after year's worth of secondary xylem that have been added initially to conduct water, but later on their leftover strength when they're dead is to provide structural integrity to the stem and make it stronger. So again, we've seen our three different tissue types, right? We've got dermal, ground, and vascular. Dermal is protective, ground is for kind of general functionality of the plant, and vascular is for transport of water and nutrients. So what we're going to do now is we're going to focus in and take a closer look at the three different tissue types and take a look at how they're produced. So we'll start off with dermal tissue. Dermal tissue is typically only a single cell layer thick, in most plants at least, and it's the outer covering which protects the layers below from things like dehydration and herbivores and drying out and all kinds of things which could cause maybe infection or something like that. Typically on the outside of the dermal layer is something called a cutin layer, and cutin is going to also have a layer of what's called a cuticle associated with it. And a cuticle is a layer of colorless, clear wax that helps to seal water into the plant so it doesn't leave too quickly. Cuticle is very, very important for preventing drying out. There can be other cells associated with dermal tissue as well. There can be guard cells, as we'll see, associated with stomata. Stomata are openings, typically on a leaf, sometimes on a stem, to allow gas exchange to occur. They're like how the plant breathes, is by opening and closing stomata. The cuticle helps waterproof the rest of the leaf so it doesn't lose too much water. Trichomes are hairs. So these little things here, this by the way is a scanning electron micrograph taken with a scanning electron microscope of trichomes on the dermal surface of a leaf. And root hairs, which kind of show up the same way but have a very, very different function, would be in the roots. They're going to have a very important function when it comes to absorption of water and nutrients in particular. These are guard cells. Guard cells are the cells that surround stomata, which are like little tiny mouths, typically on the underside of a leaf. So there are actually two cells here. One of them is like a half moon up here. The other cell is a half moon down here. And so what happens here is that when stomata are open, what occurs is these guard cells have filled up with water. And when they fill up with water, they're going to open up asymmetrically to create more or less like a hole in the leaf to allow gas exchange to occur and also to allow water loss to occur. There's some really interesting genetic things that go on here with determining how closely spaced guard cells and stomata are to each other. Basically what happens is if a certain region on a leaf has a stomata, there is a certain minimum distance you have to travel away from that before you'll see another stomata. Right? So having too many of them too close together can actually be not only expensive but counterproductive. And so plants do a really good job genetically of making sure that these cells are somewhat spaced out apart on leaves and stems. So it actually tells cells where they are in relation to other cells of the same type, and that will therefore determine what they're going to do and how they're going to function. So again, we see stomata here on the underside of a leaf. Each one of these are guard cells, and in between that is a stomata. These can be opened or closed in response to either really humid conditions when they're going to be open or dry conditions when they would tend to be closed. Now trichomes, again, these are like little fine hairs that you find on the surface of leaves and of stems. If you think about something like a tomato plant that's got like really hairy stems and really hairy leaves, they are covered up with trichomes. These things can have a lot of function. One of the things trichomes do is they'll help cool leaf surfaces by sort of providing shade almost in a way. If you have enough of these and they're really, really thick on the surface of a leaf, that's going to prevent quite so much sunlight from hitting the leaf and therefore prevent it from heating up quite as much to also minimize water loss. They can also reduce evaporation by trapping a boundary layer of air. So basically here, because there are so many trichomes, the air in and among here is going to be relatively humid, relatively dense. The air in contact with the stomata is going to be relatively humid. So even when the stomata are open, they're not going to be losing as much water because they're opening up into a more humid environment. There's also less airflow here, right? So if the air isn't moving very much, you're also going to have less evaporation. Kind of like on a really hot, humid summer day when the air isn't moving. You sweat and the sweat just kind of sits on your skin and it pools there and it doesn't really do anything to cool you off because it's not evaporating. That's kind of what this does. It's kind of the same idea. It's preventing air from moving over the surface of the leaf, and that helps to minimize evaporation, which is huge if you're a plant. Some of these can also be glandular. You can have glandular trichomes, which is what these are. They have these structures up here which can produce toxins or adhesives or feeding deterrents to keep herbivores away from the plants as well. Lots and lots of diversity in trichomes. Again, there's kind of a similar thing here like we saw with stomata, so that plants don't produce too many trichomes in the same location. If this cell here produces a trichome, which is a little outgrowth from the cell wall, this is the same cell, it's a little growth out in the environment here. What this cell is going to do is it's going to inhibit other cells around it from producing trichomes. So you might have to go 3 or 4 or 5 cells away before you'll find another cell that looks like this, because this one is inhibiting all the ones around it from doing the same thing. There's not a whole lot of point in overproducing trichomes, right? There's a certain number which will help, and beyond that point, it's diminishing returns because you're spending more and more energy and not really getting much return on that investment. All right, so we're going to go ahead and stop here. I'll pick up the next section with root hairs, and we'll go from there.